Hello guys, today we're going to talk about connective tissue. So, generally speaking, as I said before, all the tissues are divided into two different portions of the tissue, the cells and the ECM. We already discussed that some tissues, in the epithelial we'll discuss this thing, some tissues have more cells to more ECM. For example, in the case of the epithelial tissue, we have high amount of cells with very low to almost no presence of extracellular matrix. In the connective tissue, we find the exact opposite. We're going to find very few cells which is disproportional to the very high amount of ECM. So in this video, we're going to talk about first the cells, and then we're going to talk about how the cells produce the ECM and how the ECM and these cells come together to form this connective tissue. So first up, one of the most important and most prominent cells that we're going to meet in the connective tissue is the fibroblasts. The fibroblasts, which will differentiate into fibrocyte. So as the name suggests, fibroblast is the cell that produces fibers. Generally speaking, all the term blast means to produce. So in the case of these cells, we have the fibroblasts that produce fibers along with ECM. We'll discuss what type of ECM. Now these cells have these two different forms. The highly active form, which is the fibroblastic form, in which case we have a more prominent nucleus and a more widened and more enlarged cytoplasm, and when these cells tend to become quiescent or less active, way less active than their progenitor cells, then they differentiate into fibrocytes, and they have a characteristic appearance of very thin-like cells with thin processes and very shrinked nucleus. So for example, in this case, we're going to see clearly a fibrocyte as it is right here, and then a fibroblast, which is much more prominent and much more easy to distinguish, and we can see the clear cytoplasmic processes along with the large nucleus. So these cells produce, as I just said, fibers. One of the most important fibers that we were going to meet in the human body, and generally the human body, and specifically in the connective tissue, is the collagen. There are many different types of collagen, and we'll discuss in the collagen later on along with the rest of the fibers. So they produce the fibers of collagen, also reticular and elastic in some cases. We'll see when. Elastic not so much, typically reticular and collagen. Now they produce other parts of the ECM, such as glucosaminoglycans, proteoglycans, multi-glycoproteins, and growth factors. We'll discuss all these contents of the ECM in detail next on, but very quickly, the glucosaminoglycans along with the proteoglycans create the scaffold and the matrix in which the connective tissue is going to be produced and going to be formed. So this is practically these components along with the glycoproteins are the components that structure and practically put the pillars of where the connective tissue is going to grow and how it is going to be grown in and what it's going to grow into. So fibroblasts again are typical cells that are mesenchymal origin, and in some cases, some pathological cases of injuries, then these cells will transist and will differentiate into myofibroblasts. These are cells that have both features of fiber producing cells along with contractile elements. This is, for example, why in scar tissues, in the scar after a deep injury, we can find this, let's say, this contracted image, this shrinked image of the trauma, and this is because of the presence of the myofibroblasts, because they both produce the collagen that's going to be the scar tissue part of it, and along with and also this contractile appearance is going to be because of these unique cells. So next up, we're going to find adipose cells, fat cells. Now typically there are two types of these cells, the white adipose tissue and the brown adipose tissue, unilocular and multilocular. There are two synonymous different terms. Of course, there have been recent papers that have suggested and proven the existence of white and in between brown, and they're called yellow adipose tissue, but this is beyond the basics of what the point of the video. So in this case, we're going to find the unilocular and the multilocular. Both of these cells, as of course with all of the sources of them, cells of connective tissue, are mesenchymal stem cells, come from the mesenchyme. So the mesenchymal stem cell can differentiate, for example, according to the scheme, to fibroblast along with many other cells, and also to adipocyte. This is very interesting to underline. We should underline that before you have the differentiation of the mesenchymal stem cell to the adipose tissue. There is this in the transient stage, this in-between stage between the final last stage of either multilocular or unilocular, and this is going to be called pre-adipocyte or differentiating adipocyte. This is interesting. I'll exactly explain why is it interesting in just a second. So we have the two different tissues, of course, the white and the brown. The differences, along of course with the morphology, is that in the case of the unilocular, and this is called because what this has one very wide, very big vacuole of lipid, and this is why it's called unilocular, only one locus. And in this appearance, of course, in this cell is responsible for storage of energy, storage of triglycerides, storage of any fat forms of the human body. And of course, they play a very important metabolic role when it comes to the function generally of the hormonal system, along with the metabolic system and the GIT function as well. So because of this very wide vacuole inside the cell, of the fat vacuole inside the cell, we're going to have this typical appearance of the punched out nucleus in the peripheral placement of the nucleus, as we can see both in the schematic and in the histological slide. So the function again is typically metabolic in this case. Metabolic to maintain fat, to maintain storage, to maintain energy, and as a consequence, because of the nature of fat, is insulation, mechanical and thermal insulation. It means that it simply stops and halts any thermal loss. For example, this is why like typically we have excessive amount of fat in the tissues in the skin, prevents heat loss, and it's more opt for survival in colder climates. So this is the one function of this is the one part of the adipose tissue. The second part is the brown adipose tissue, the multilocular. In this case, there are many small vacuoles of lipids inside the cell, but whatsoever the function of the brown adipose tissue is almost completely different than the white adipose tissue, and this is because these cells have unique functions. In fact, the adult human body contains almost no brown adipose tissue. Typically, we can find in infants and in hibernating species, and this is because the function of the brown adipose tissue is not to store fats only, but primarily it is to produce heat. It is one of the cells that is responsible for thermogenesis. In this case, these cells are unique, of course, based on their function, but also based on their structure. Meaning what? The mitochondria, typically, as we know, they produce ATP, and this ATP is used for many of the cell functions. In this case, the mitochondria of the brown adipose tissue do not produce any ATP whatsoever, zero. The whole electrochemical gradient between the mitochondrial membrane and the two different sides of the electrochemical gradient is used to produce heat. And in this case, the mitochondria contain unique proteins called thermogenins, and the name is perfect to describe exactly what it is. Thermo, again heat, and genis, to produce. So this is exactly the protein that produces heat using this difference of the H protons, the difference in the protons in between the two different membranes of the mitochondrial membrane. So again, the function here is thermogenesis. Now, before I mentioned that it's important to underline the differentiating adipocyte, the first stage before the differentiation to the white or the brown adipose tissue. And this is important because there is this transition, of course, in the infant, in our infant lives. We have brown adipose tissue, typically located in the neck or shoulder. Generally, very few areas of the infants have brown adipose tissue. And this tissue, of course, does not exist as much. It's vastly and greatly reduced towards our adult life. And this is because there's a transition from brown adipose tissue to white. And how this procedure is happening is because the brown adipose tissue is de-differentiating to this the pre-adipocyte or the differentiating adipocyte, and then transitioning again, differentiating again into the white adipose tissue. Of course, this process can happen both ways, from brown to white and from white to brown. Typically, it's not seen the white to brown in the humans, but it's also a possibility. In vitro, it's possible to be done. So And this is, of course, a very, very characteristic feature of the high amount of round adipose tissue in hibernating species. This is because, such as bears and snakes and other animals that sleep through the winter and wake up in the summer. And this is because in these cases of the hard winter, these animals need to produce heat to maintain, of course, to stay alive. And this is a typical feature of these animals. So the morphology, of course, is again this of the brown adipose tissue is filled with mitochondria, again to serve its function. And we're not going to typically see brown adipose tissue in the adult human body, so the histological picture is not really important in this case. So let's move on to one of the greatest and most important, let's say, groups of cells of the connective tissue. So we have the mononuclear phagocyte system. This is, in short, I'm sure you've heard the term macrophage, and I'm sure you've seen the cells over and over again in biology and many other sources. This is a primary immune cell. This immune cell is responsible for phagocytosis along with many other functions. It is one of the first lines of defense in the immune reactions. And the reason why we call it mononuclear phagocyte system and not just macrophages is because the monocytes, again, the monocytes are the precursor cells of the macrophages. So the unit of this system is the monocyte. Of course, the monocytes can actually group together, form to different locations, go into different locations, form different structures, they're so flexible cells, and they have a very wide variety of functions, and this is why we call it a system, because we can see the many different forms of the monocytes within the system and performing almost similar functions in all the cases. Of course, there is a very nice table that can help prove the point that I'm discussing right now. Of course, it's von Jonquer. So here we find the monocyte. The monocyte again are the primary cells that are being produced in this system, in the mononuclear phagocyte system. So the location of the monocyte is in the blood. Of course, these cells, as I said before, are the precursor cells of macrophages. So whenever a monocyte goes from the blood, from the mainstream to a specific tissue, then we have the procedure of diapedesis, and in this case, this monocyte is going to differentiate it to a macrophage. So the macrophage, of course, is responsible for phagocytosis, production of cytokines, chemotactic factors. It's one of the biggest orchestrators and one of the biggest maestros of the immune defense, both primary and secondary, both innate and adapt immune system. So whenever we have the monocytes being placed in the liver, they're going to be called Kupfer cells, and this is because they perform unique functions. Generally speaking, whenever we have a different term, for the same cell, then typically this cell in this location performs some unique small little function extra to that of the original cell, typically speaking. So in the liver, we have the macrophages, they're going to be called Kupfer cells. If we have in the CNS, we're going to call them macroglial cells. Langerhans in the skin, dendritic in the lymph nodes, and so on and so on. And here we find one unique characteristic of the human body, that in a physiological condition, we have the fusion of many macrophages to form the osteoclast. This is the bone. This is found in the bone, and there's going to be an extensive talk about the osteoclast in the video for the bone. So let's move on. Now mast cells, also a very important group of the connective tissue. The mast cells are primarily known for the allergy reactions, but this is not necessarily 100% true. Of course, this is a valid point, but the mast cells have a very wide range of functions. One of which, and most clinically important, yes, that's true, most clinically important is the allergic reaction, or the allergic shock in the worst case scenario. How does it happen? How does this cell work, and why do we need to know things about this cell? Well, because the mast cell produces a wide range of molecules, but two of the most important we're going to talk about right now is heparin, which is actually also used therapeutically in cases of hypercoagulative states, in which case the patient's blood is too, let's say, too dense for many reasons. This is a discussion that is better discussed in pathology and not in histology, but the point is that it functions as an anticoagulant. So it practically prevents the blood from coagulating and from stabilizing a blood clot. And the second and very important feature is histamine. We have the production of histamine by mast cells. What is histamine? Histamine is a specific molecule that induces two functions, very important functions. First off, it increases vascular permeability, and the second is smooth muscle contraction. So what's the big deal about it? Well, the thing is that imagine a patient that has an allergic reaction, a severe allergic reaction. The first thing we're going to see in this patient is, of course, the hives, which is practically many red spots on the skin, and this is because of the increased amount of histamine, which increases the vascular permeability, which practically forms a vasodilation of the capillaries of the skin, and this is why we see the red color. It's because we have the vasodilation on the skin, and we can see the dilated blood practically inside on the skin surface. And the second and much more important, more possibly fatal reaction and action of the histamine is the smooth muscle contraction, because our respiratory system is filled with smooth muscles. So whenever we have a trachea which is covered in smooth muscle, which contains high amount of smooth muscles, in this case, when there's a high amount of histamine in the blood, of course, there's going to be a high function of contraction in the trachea, and this is why we see patients that undergo allergic shock reaction that they cannot breathe. It's because the trachea is closing up, and the conducting portion of the respiratory system does not allow air to go by, because the conducting portion is again filled with smooth muscles. So, parenthesis, of course, the very important thing to do is when you recognize this patient is, of course, give him adrenaline to save his life. And these are the very two different, very important products of mast cells. Now, if we actually see mast cells in a wider range, in a more, let's say, in the general picture, we can see that these cells are actually orchestrators of many different functions. They can be pro-inflammatory. They can induce pro-inflammatory markings. They can inhibit inflammatory markings. They're anti-inflammatory functions, pro-inflammatory functions, reparatory functions. So, and this is seen because this is, for example, one table that contains the whole range of the contents of the products of mast cells, and you can see there's a very wide range cells that are filled with enzymes and proteins of destruction, of reparation, of inflammation, pro-inflammation and anti-inflammatory proteins. So keep in mind that these cells have a much wider role, and they're typically found in the perivascular space surrounding a vessel. One unique feature of the mast cells is this metachromasia. This is the staining characteristic, that they can be stained with eutolidine blue, for example, and they have a very unique granular appearance in the microscope. This is, for example, the appearance of a mast cell in the microscope under toluidine blue. So let's move on. Before we move on, I want to underline one thing. There are two steps to allergy, generally speaking. This is, of course, a topic best discussed in immunology, but just a short mentioning right now. First, we have the IgE receptors. This is the antibody specifically for the antigen, the allergen, of course, the molecule and the substance or foreign particle that induces the allergic reaction. So whenever you have, let's say, pollen, typically pollen is a very common allergen for many people. So whenever a person inhales pollen for the first time, then there's going to be this whole reaction, immune reaction, that produces this IgE, these unique antibodies against pollen. The first time this person is going to be exposed to pollen, there's going to be no reaction. Now these antibodies that are being produced against pollen are going to be anchored on the cell membrane of mast cells. So in this case, in the second appearance of pollen, then pollen is going to act as an antigen, is going to attach on the antibody of the cell membrane of the mast cell and induce the degranulation. These mast cells, of course, have the two functions of degranulation, which is they contain all of these granules that contain the wide range of products that we discussed just earlier before, They place them in granules and then they exocytose them upon activation. And there's of course the regranulation, which is the reproduction of these granules. But to come back to the point, after the second time of the human body receives pollen, the stimulus of pollen, then it's going to be the activation of the IgE and the degranulation is going to take place.